RNA extraction. Before beginning, clean the working area with ethanol 70% and 3% hydrogen peroxide by atomizer. Add 1 milliliter trizol solution to the tissue, then homogenize the sample using homogenizer. Then incubate the sample for 5 minutes at room temperature. Transfer the tissue homogenate to a new Eppendorf tube. Add 200 microliters of chloroform to the sample. Close the cap of the tube, then make inversion twice. Incubate the sample for 3 minutes at room temperature. Place the sample in the centrifuge for 15 minutes at 12,000 RPM and 4 degrees Celsius. By the end of the centrifugation, you'll be able to see upper colorless aqueous phase and interphase and lower red organic phase. Transfer the aqueous layer carefully to a new tube. Store the interphase and organic phase at 4 degrees Celsius in order to use them to isolate DNA or protein. Add 500 microliters of isopropanol to the sample. Close the cap of the tube, then make inversion twice. Incubate the sample for 10 minutes at room temperature. Place the sample in the centrifuge for 15 minutes at 12,000 RPM and 4 degrees Celsius. By the end of centrifugation, you'll be able to see total RNA precipitate as a white pellet at the bottom of the tube. Remove the supernatant using pipette. Wash the pellet with 1 milliliter of 70% ethanol. Close the cap of the tube, then make inversion twice. Place the sample in the centrifuge for 5 minutes at 7,500 RPM and 4 degrees Celsius. The ethanol supernatant is removed using micropipette, then leave the RNA to dry in air for 5 minutes. Suspend the pellet in TE buffer by adding 50 microliters of RNase-free TE buffer, then make pipetting. Incubate the pellet in water bath at 60 degrees Celsius for 15 minutes. Now you have RNA sample ready for use in any other processes.